Hello and welcome to the lecture on RNA structure prediction. The reading is Xiong chapter 16. And today we are going to be talking about the different types of RNA structures, the building blocks of RNA, the loops, things like that. And then we're going to go on to secondary structure prediction methods. These are two broad classes: the ab initio approach, meaning based on a single sequence, and the coevolutionary approach, which can be used when you have a multiple sequence alignment, and how to evaluate these methods. And then we'll go on to tertiary structure prediction, or 3D structure prediction. And namely, this will be homology modeling as well as integrative modeling. So, RNA structure prediction. So, this is not an easy task. The 3D structure of RNA is hard to predict for the same reasons that RNA is hard to crystallize, and it has to do with the charge. The backbone has a negative charge. Each oxygen has one of the two oxygens has a negative charge on the phosphate. It has more flexibility in some sense than protein. It tends to be bigger in terms of each residue of a nucleic acid is much larger than a residue of a polypeptide, and it's sticky in a sense. The charged backbone makes lots of cool interactions. The bases, especially adenine, have lots of ways to make hydrogen bonds and stacking interactions, and it tends to stick a lot. They tend to fall into kinetic traps for this reason, and they take longer to fold. Okay, so there are basically three main ways you find RNA. One is mRNA. So as you know from the central dogma, DNA gets transcribed into mRNA, and that from there can be translated into protein. There is tRNA, and so as you recall, the tRNA reads the one codon of mRNA in the ribosome. And lastly, there's ribosomal RNA, what belongs to the ribosome proper. And so mRNA is once it's ready to be translated, you can think of it as mostly unstructured, but then there are such things as introns, and these can have some sort of structure. It can be quite large and complex, actually. And of course, the introns get cleaved out. It's only the exons that remain. tRNA, you may recall, it's kind of very roughly looks like this, and it's got an anticodon over here which reads the mRNA, and over here it'll if it's charged, it'll be carrying some sort of amino acid. And then lastly, there's ribosomal RNA, and that's the RNA that's in the ribosome. In the case of prokaryotes, you're familiar with 16S and 23S RNA. And that accounts for, so three main classes of RNA. And of course, there are others, but they're smaller in quantity. So what do we mean by primary, secondary, and tertiary structure? I think we covered this when we did proteins, but anyway, primary structure just means basically a sequence, whatever that may be. Secondary structure typically refers to just the helices. So if you've seen secondary structure diagrams, they basically tell you where the helices are, and everything else is kind of looks like a loop. But tertiary structure is when you take into account all the contacts in the RNA or DNA, and build up a 3D structure, right? So this could be this would include, of course, still include the helices, but which have canonical Watson-Crick interactions. But then it also includes these additional interactions which may be of arbitrary type, like that may include non-canonical base pairing interactions, using all three of their possible edges, and even edges even interactions that are not described as base pairing, salt bridges and so on. So the building blocks of nucleic acids you should be familiar with by now. So in the case of RNA, you have uracil. In the case of DNA, you have thymidine. And we're going to talk about how this has a backbone, right? C5 prime. It's got a reasonably intuitive numbering system. Remember, it's got a charged backbone. There's one of these two oxygens carrying negative charge. Let's see, we got a C3 prime, C2 prime, C1 prime, O4 prime. In the case of RNA, we have an O2 prime and an H. And over here we have a O3 prime and then a phosphorus. So you don't need to memorize the names of the atoms on the bases, but you should memorize the naming convention for the atoms in the backbone, including the ribose. And it's very simple. Like, you start with C1 prime, which is here where the chi bond starts, where the glycosidic bond starts, followed by C2 prime, followed by C3 prime, followed by C4 prime, and then up here is C5 prime. And then this oxygen in the ribose is conventionally named after the C4 prime, O4 prime. And then these atoms that branch off from one of these ring atoms, they follow the same numbering, or they adopt the numbering of the atom they're attached to. So this O2 prime is named after the C2 prime, and this O3 prime is named after the C3 prime. Okay. And of course, you should know that uracil and cytosine are pyrimidines, and adenine and guanine are purines, right? And in a second, we'll talk a little bit about how these can interact with each other in canonical and non-canonical base pairs. So a nucleic acid residue has three sort of so-called edges under the Leontis and Westhof nomenclature. Leontis Westhof. These edges are the Watson-Crick edge, which is the one that you're familiar with from nucleic acid helices. There's a Hochstein edge, which is sort of on this far side of this base. And then there's a sugar edge, which is called that because it involves this O2 prime oxygen, which is actually on the sugar ring, not on the base. And so the idea beyond this nomenclature is that any of these edges can interact with any of the edges on another residue, right? So if you have some other residue over here, this Watson-Crick interaction, for instance, could be interacting with this Hochstein edge of this other base. This was set down in the Leontis and Westhof catalog in their RNA paper, 2001. So here again, they're sort of indicating the Watson-Crick edge, the Hochstein edge, and the sugar edge. And then in addition, there's the so-called orientation. And so the orientation means that which way the glycosidic bonds are pointing. So if you draw an arrow pointing from the glycosidic nitrogen to the C1 prime carbon on the ribose, let's say it's pointing down. If the other one is also pointing in the same direction, then we say that's in cis. But if they're pointing in opposite directions, see here, this one's up and this one is down, then it's in trans, right? Or you can think of it as saying, okay, which side is the ribose on relative to the hydrogen bonds, these hydrogen bonds in the middle. If it's the same side, then it's cis, and if it's opposite side, it's trans. So cis is just Latin for on this side, and trans is Latin for on that side. Okay, so I said that any base or any one base can use any of its edges to, in principle, interact with any of the edges on a second base, and it could have either cis or trans orientation. In principle, there are some combinations that are not stable. But here are some examples. So let's say this adenine is using its Watson-Crick edge here to interact with the sugar edge of a second adenine, and the glycosidic bonds are in cis. They're both in the same, pointing in the same direction. So we would call that a Watson-Crick sugar edge cis. Here's an example of a Hochstein sugar edge. Let's see. So here's the Hochstein edge, and then here's a sugar edge. And think for yourself, what orientation are the glycosidic bonds in? Cis or trans? So the answer is cis. Actually, I mean, it says so right here, of course, but it's a good mental exercise to try to ask yourself if you understood which way it works. So this one is a sugar edge interaction with another sugar edge. So sugar edge, and the glycosidic bonds are again in cis. Let's see. So last one might be this. This is a Watson-Crick sugar edge interaction, and this one is actually in trans. So if you see here are the hydrogen bonds, and this glycosidic bond is pointing kind of that way, and this one is pointing the other way, and so that's trans. I'm going to delete those lines so you can see it a little bit better. And oops, I'm sorry, I put my picture on top of the last two over here. Let's move it temporarily over to one side. And here you see examples of that would be a Hochstein here with a sugar edge there, and then here's a Watson-Crick with a sugar edge, and all three in this bottom row are of course trans, as stated there. Okay. So the Leontis Westhof nomenclature is useful again for specifying the orientation of one base with respect to another. And these are in general not bases that are consecutive in sequence. They're bases that are base paired, and they could be very far apart, but they have to be most likely at least 3 residues apart. Depending on what interaction it is, maybe it could be less. But at any rate, it's not in general residues that are consecutive in sequence. So for residues that are consecutive in sequence, there's a different structural alphabet, which is complementary to Leontis Westhof catalog. And this was developed by Bohdan Schneider, a friend of mine in the Czech Academy of Sciences, and also by Gina Chebny, who's also a friend and collaborator. And so the way that the nucleotide conformers work is they specify the conformation mostly of the backbone of a dinucleotide. So here's a picture of a dinucleotide, and you can see here they've labeled all of these dihedral angles along the backbone. And a given dinucleotide, let's say one that's called AA00, would specify what's the dihedral angle delta 2, what's gamma 2, what's beta 2, alpha 2, blah, blah, blah. All of these dihedrals would be specified. In addition, there would be a couple of interatomic distances here that are specified. And as it turns out, that if you use the Leontis and Westhof base pairs and the nucleotide conformers together, you can get really good reproduction of your recapitulation of your 3D structure. Yeah, so if you recall, the dihedral angle means if you have some atoms A, B, C, D, dihedral angle is the angle you'd see if you're, let's say this is your eyeball, looking down this axis between B and C, and it's the angle that's formed between A and D. So if you're at this eyeball that I drew here, you would see A here, you would see B here, C would be kind of hidden behind this B, and then D would be over here, right? And so this angle would be your dihedral. Just to recap. So both the Leontis and Westhof base pairs and the nucleotide conformers are enforceable in MMB, a program that I wrote. And actually the nucleotide conformer part is very new. Bodan and GD and myself have been working on it for the past couple of years. Anyway, you'll play with that in the lab a bit. So in terms of what RNA looks like, there are such things as loops. And so they have this sort of naming convention where a loop at the end of a helix is called a hairpin loop. And why is it called that? Well, you know what a hairpin looks like. Kind of does this, right? So you can see the resemblance. And a bulge is a loop that involves only one strand of the nucleic acid. We're usually more talking about RNA, but it could happen to DNA also. Anyway, so all the bases in the other strand are paired, but this one has one or more bases that are not paired. This strand. So that's a bulge, and then an interior loop is different. Here you have both strands have bases that are not engaged in Watson-Crick interactions. Actually, I should have said that they're not paired. They're just not paired in a Watson-Crick fashion. They could have other non-canonical base pairings. So that's an interior bulge that involves two loops. And then there is a multi-way loop or a multi-way junction, and that means more than two helices are coming together at some point, like this. Okay, so we said that there's a way to predict secondary structure ab initio, and again, ab initio means in this case from primary sequence only, not using multiple sequences. And so one simple way to do this is with a dot matrix. And the way the dot matrix works is you have the same sequence here on one axis and on the other, and you're kind of in a sense aligning it against itself, and you're looking for reverse complements. So for instance, here this G can form a Watson-Crick base pair with this C, right? That's the same thing as saying that this G is interacting with this C over here. You don't actually need to draw the other half matrix. Just the upper well, either one, the upper or lower triangle are fine. And so this A, you can also see that it could make a base pair with this U, and this C could interact with this G, and this C could interact with this G here, right? But in principle, if this were a different sequence, let's say longer, then it might have additional diagonals, somewhere, and some of them might be contradictory, you wouldn't be able to form all of these diagonals simultaneously. But anyway, you can see how this looks a whole lot like your alignment matrices that you looked at earlier for pairwise sequence alignment. So you could apply the same sort of dynamic programming, and then find ways to make a gapped alignment that connects multiple diagonals. Okay, so in this case, so you see here the final results, but if you're following this plot, you could just say to yourself, okay, well, if I want to draw a secondary structure diagram, I would write A, and then I'm going to get into this helical part, CUGG. So let's draw it like this, CUGG, and then there's going to be a CCAG part, which is also part of that helix. And there's this stuff in the middle, which is just going to be a hairpin. So it's going to look like AUA, CG, and then the rest of the helix. So it's CCAG, AA. Right? And so it's kind of a good skill to be able to interpret these dot plots, and in general be able to sort of write down secondary structure in a couple of different ways. Like here I translated dot plot into this secondary structure diagram. Okay. So one ab initio method is called MFold. There's also a very similar method called RNA fold. Anyway, the way MFold works is it tries to predict secondary structure based on energetics as compiled in these so-called Turner parameters. And so what the Turner parameters tell you is things like, okay, how much energy is gained by forming a base pair, right? So this GC might give you some score, right? It's GC, as you know, is stronger than AU. So whatever the score this gives you, you'd expect it to be higher than the score that you get for this UA, right? So anyway, so you get some points for forming this GC, just GC, and this GC down here, and some different points for making this UA. And then on top of that, there's some energetic benefit to stacking these two consecutive bases, actually on both sides, right? And it depends on the residue, the densities of those residues, how much energetic benefit you get. Anyway, tabulate that. And then you have the entropic cost of closing this loop, right? And that cost will be higher the longer the loop is. And there's a table for that. And then lastly, there's this sort of an entropic cost for this tail becoming shorter. Like now this free end is only 2 residues, before it was longer than that. And so you pay an entropic price for that. There's a way to just sort of add up all these energies using dynamic programming. And it's implemented in this program called MFold and 1 or 2 successors thereof. Now, if you happen to have a multiple sequence alignment, which is probably not hard to get depending on what your situation is, then you can do something called coevolution. And I believe we already talked about this in an earlier lecture. The way coevolution works is if you have some sort of multiple sequence alignment. So let's say here we have this one that's AGCU, and there's another one called UGCA, and then there's UCGA. I'm just taking these 3 and piling them on top of each other. And if you notice, or if you didn't even look at these structures that you have below you, you might notice that if you look at position 1, I'm just going to call this position 4. Whenever you have a U here, you have an A here. See? And then here you have U again, A again. And conversely, when you have an A here, you have a U here. Right? So every time position 1 changes, position 2 has to change. And always in the same way. Or at least maybe you don't really believe that yet because you only seen 3 pairs. But then you look at position 2 and 3, and you see, okay, when there's a C here, there's a G there. When this changes to a G, this changes to C, and this is maintained here, G goes to C. And even that might not be enough to really convince you that there's a pattern, but anyway, the idea is that if you see lots of lots of sequences, like in a bigger non-cartoon example, you should be able to show whether these are changing in a coordinated fashion. And the way one way to compute this, that's sort of the most intuitive way, and the way that's most sort of firmly rooted in information theory, is by computing the mutual information. And mutual information basically just tells you how much one variable knows about another variable, right? So if your residue at position 1 knows in some sense about the residue at position 4 or vice versa, then you say that they have mutual information. And mutual information quantitatively means that if, let's say, this is position 1, this has this much information, and position 2 has this much information, this would be the mutual information. How much of position 1's information is known by position 2. And in this case, if the coevolution is perfect, then actually your Venn diagram would look like this, right? Your mutual information is basically 100% of position 1's information as well as position 2's information. Right? In a more practical situation, you wouldn't get quite such a strong signal, most likely. But nonetheless, this is a really powerful method. In fact, something like 90% of RNA secondary structures are predicted by mutual information. In fact, the secondary structure of tRNA was predicted by Mike Levitt back in the 70s using 14 sequences. And many years later, I think maybe it was in the 90s, Robin Guttel did the same calculation again using thousands of sequences. And guess what the difference was? He found one additional base pair. So for RNA, mutual information is really powerful. It's more powerful than for proteins, because for RNA you have a smaller alphabet, 4 residues as opposed to 20, and also the interactions are more modular. Okay, but anyway, that's sort of a bit of an aside. The way you would actually compute this is with this formula here. So P of Pxy is of x comma y. That's the joint probability of x and y, and P sub x of x is the marginal probability of x. P of y sub of y is the marginal probability of y. So let's say we were just to compute this for position, again, 1 and 4. The way you would do this is you would say, okay, I'm going to sum over all possibilities. So for example, there's a P of U comma A. That's going to be 2/3. And then P of A comma U, that's 1/3. And notice we don't do GC because we do this on the basis of just what we see in those 2 columns, right? You don't need to take into account the other columns. So then you need the marginal probabilities, P of U, and this is the P of U at position 1. You don't include. Don't mix that up with the probability of U at position 4, because in principle these 2 variables could be encoded in completely different alphabets, right? I mean, you could have RNA residues on one side and something else on the other side. Like the other object could be some completely different data type. So it doesn't have, they don't have to use the same sort of they don't have to be similar variables in a sense. They kind of have to be discrete, but there's kind of ways you could even deal with continuous data. Anyway, so you want P of U at position 1, and that's 2/3, and you want P of A at position 1, and that's 1/3. And the P of U at position 2 is kind of the converse of that, or the complement of that. So that's 1/3, and position 4 I meant. Position 4, A. equals 2/3. Okay, so basically to add these up, what you do is you say, okay, first let's do P, this first thing was P of UA, and so that's 2/3. Log 2 of 2/3 divided by P1 of U, which is 2/3, times P4 of A, which is 2/3. Right, so you just eliminate one of these 2/3 and it becomes 3/2. So 2/3, so something like 2/3 log of 3/2. And then the second thing is, the second term is P of AU, which is 1/3 log of 1/3 divided by P of A position 1, which is 1/3, and P of U at position 4, which is also 1/3. Okay, so here again you cancel the 1/3, and so you get the log of 3. So 1/3 log of 3. So you can easily see that, or I mean, just do this calculation if you want on a piece of paper, and you'll see that this is some number that's greater than 0. So these variables do have mutual information. So you can say that they're coevolving. Okay. All right. So those are the two main ways to predict secondary structure. What about tertiary structure? So if you have a known homolog that's been crystallized or otherwise resolved, then you could do homology modeling. And I wrote the first method for homology modeling of RNA. And the way it works is you have some template, like say this tort, and then you have some target, which is your RNA of unknown structure. And what you do is you find corresponding atoms or corresponding residues by doing a pairwise sequence alignment, and you just attach them to each other, basically with springs between corresponding atoms. And then you let the dynamics run for a while, and it superimposes your target on your template. And it's a relatively straightforward process. And now there are online services that do things like this. For example, Moderna, which is written by my colleague of mine, Janusz Bunici in Poland. Yep. And then lastly, there's integrative modeling. And the way it works is you can specify all the base pairing interactions between the residues in your RNA. So let's say you did a secondary structure prediction, so you know that residue 1 is interacting with residue 4, 5, 6, 7, 8, 9, 10, 11. You create a Watson-Crick base pair, and then likewise another Watson-Crick base pair between residue 2 and 10, and so on. And that gives you the helix, but this loop is still kind of loose, and you don't know what it's doing. Then it could be that even within this loop, you know the non-canonical interactions, right? So you might say, oh, well, there's some sort of like, let's say Hochstein sugar edge interaction between these two residues, just to say something. And then lastly, you might know what sort of nucleotide conformers you should expect. So for example, here this helix, if it's an A-form helix, then you could expect something like AA00, which is the helical nucleotide conformer. And then if you say like this is a GNRA, which is one of the motifs for tetraloops, then you might know some of the NtCs within the tetraloop. And you put these pieces of information together, and you can get a really good 3D model of your structure. Sometimes, depending on what you have, sometimes maybe even less than 0.2 angstroms. I'm not kidding, or around 0.2 angstroms. And then integrative modeling, of course, means that other information that you might have, you can also integrate. I mean, you might have like a very low resolution cryo-EM density. You could use that. You might have some distance constraints, let's say from NMR, and so on. So that's about all I wanted to tell you about RNA secondary and tertiary structure. And I will see you when you come in for the discussion section. Thank you.